Welcome to ASM Live, the talk show live from the American Society for Microbiology 2013 General Meeting in Denver, Colorado. This talk show focuses on all things microbiology, interesting things from this meeting. I'm your host. I'm Stanley Malloy. I'm the Dean of the College of Sciences at San Diego State University and the chair of the editorial board of Microbe Magazine. I want to first begin by encouraging people in our live audience here to ask questions today. Please use the microphone if you have any questions so that all of the people listening online can hear your questions as well. And if you're listening in online, please send us questions through Twitter at #ASMLive. So in this session, we're going to focus on organisms from extreme environments and extreme things those organisms do. And this is stimulated by a session that Amy Vollmer is running this afternoon. Amy is a professor at Swarthmore College. Would you tell us a little bit about this session, Amy?
Sure, Stanley. I'm delighted to convene a session that reflects the true diversity of microbes in our environment. The session's name is Extraordinary and Extreme Microbial Lifestyles. And in it, I think it captures all the different places that we can find microbes on this planet. And basically, the common denominator is there has to be some form of water, but any form of water, ice, liquid, or vapor. And as a session chair, I got to choose from a number of abstracts submitted by graduate students and postdocs, a talk that might be of interest in this session. And I chose an abstract submitted by Natasha, who is a graduate student in Costas' laboratory, and he is one of two PIs exploring the troposphere, the clouds. And so I think we'd like to hear more about that project from them.
Before we do that, let me give a little bit more of an introduction. So Natasha is Natasha de Leon Rodriguez, and Costas is Costas Constantinidis. And they are at the Georgia Institute of Technology in Atlanta. Why don't you begin and tell us a little bit about what you've done and why we should be so excited?
So what we've been doing is we're sampling the upper troposphere to see what kind of microbes can survive at this type of altitudes. We're talking about 10 kilometers up in the atmosphere. And at these altitudes, you can start finding clouds and different kind of like conditions that may be detrimental for the microbes that are here in the air. But no, we found that over 60% are alive. And they have like mechanisms to form clouds. So we're interested in see how these microbes can influence the formation of clouds and the chemistry in the atmosphere.
So Costas, you're a PI on this grant. How did you get into this type of a question?
Right. So, we are interested in what microbes are doing in the environment. My group studies microbes in different environments, and it was an opportunity with my co-PI on the research, Professor Nenes, who is also at Georgia Tech. And so Professor Nenes is an atmospheric chemist. And I'm a microbiologist by training. And he had a campaign together with NASA to sample basically the upper troposphere, like 8 to 10 kilometers high, during the hurricanes. And then he came to us and said, Costas, are you interested to take some biofilters? I mean, they are interested more how clouds are forming, how they change with the climate change and things like that. I thought that was a great opportunity. I wasn't expect necessarily we will find a lot of microbes. And I think that's one of the first exciting line of research here, that at these high altitudes, above the seas, above the oceans, we see a lot of microbes, like not a lot compared to soil or the human gut. I mean, still the numbers are much lower, but we find on average about 1,000 to 10,000 cells per cubic meter. And that's quite a lot. And so that's the first exciting line of results from this research. And as Natasha said, one of our next objectives is now to figure out, do they really contribute to cloud formation or not? Because in order to have a cloud forming, you need a nuclei. You need something. I mean, water is vapor in the atmosphere, and you need a nuclei to basically condense the water on the nuclei. And so far people are thinking that this is mostly inorganic particles like sea salt, dust, etc. And now this work may suggest that actually bacteria are an important part of that. And so what we want to do next is try to quantify this relationship and try to put it in the models of how the atmosphere works. So I think there is a lot of excitement, but I need to make it clear, I think we are only the beginning, so to speak.
So Natasha, could you tell us something about what the higher atmosphere is like relative to somewhat lower?
So in the high atmosphere, like in the area that we sample, the temperature is like around minus 32 degrees Celsius. And of course, in the lower troposphere, we find like more warm temperatures that are not as cold as that. We also find a lot, the pressure is a lot higher too. So we're talking about like high pressure, low temperature, a lot like high humidity. So it's more like oxidative environment too. So it's like very harsh compared to the lower troposphere that the pressure is less.
Presumably a lot more UV
Yeah, a lot
with
of
the higher atmosphere.
A lot
right?
of more UV, of course, and
Right.
more
All-
oxidative agents too.
All of these things making it a very extreme environment.
We've learned so much about how microbes can adapt and specialize in these extreme environments. And I'm excited because I think you're on the verge of discovering even more new things. It's not just the low temperature and the high pressure and the UV, but there's going to be osmotic challenges because there's not as many salts and nutrients. How these microbes make a living, what they use as their carbon sources, what they do with electron sources, and where they dump their wastes will be really interesting. And the whole idea of microbes maybe influencing cloud formation reminds us that, you know, when they make artificial snow on ski slopes, what seeds those snow particles is bacteria that they throw into the air. So it would not be surprising at all. And I think it also reminds us the impact that microbes have on the profile of the entire planet, not only in the oceans, not only in the composition of the Earth's crust, but now even perhaps the weather. So I think it's exciting, and I think for people who come to the session, you may be able to look back and say, wow, I was there when we were first hearing about this. It's very exciting.
I don't think the ski resorts want everyone to know that they're spewing microbes. all
Too
over
late.
the place.
They're good microbes.
They're good microbes.
So one of the things that you focused on in this discussion is the hurricanes. And of course, that's because that's an exciting time to do the monitoring. But when you think about what microbes are there, the question is, what's it like when there's a hurricane potentially stirring a lot of things up versus pre-hurricane?
That's it, do you want to?
Yeah. I would say that before the hurricanes, like the composition of the atmosphere was like, I don't know, undisturbed or some kind of thing. But then after the hurricane, we saw like there was a change in the number of microbes that we found, and also the composition, since you have a lot of rain there's like all the updraft coming, like bringing all the microbes into the atmosphere, but then you also have all the rain bringing them down and like throwing all these organisms back into the earth. So you have like all these disturbance of like some of them going up and surviving this harsh condition, and then the other ones going down.
We have a question from the audience.
Hi, I actually have a couple fairly simple questions. So just to start out with, how are you collecting these samples? And then how are you determining what microbes are there? Are you culturing things or just going straight to sequencing? And then finally, what kind of micro or diversity are you seeing within your samples that you're collecting?
That's a great question. So the way we collect the samples, we have cellulose nitrate filters that we just put in the aircraft that the NASA is using to sample these different altitudes, and we just pull air through these filters, and we collect the particles that are in the filters, and then we extract the DNA from the samples, and we use 16S spiral sequencing analysis to know which organisms are there. So we just use next generation sequencing to understand this diversity. And the microbes that we found, we compared them to different microbes from different environments, and we saw that mostly these organisms were coming from aquatic environments, which make a little bit of sense since we're sampling hurricanes, and we know that hurricanes are bringing a lot of water into the atmosphere. And we also found like 17 group of organisms that are present in all our samples, independent of where we collect the samples, if it was in the continent, the ocean, hurricanes. And some of these organisms are not to utilize things that are in the atmosphere, which was quite interesting, like dimethyl sulfone and oxalic acid and things like that. So it was a quite diverse community.
Did you find anything that had not been discovered before? Anything new?
There's a few, as always, 16S sequences that are not similar to any other sequence in the environment, but in the database, but we still need to do more research in that area.
So those 17 that are pretty constant are really exciting because they kind of describe really what the constant microbiome of that environment is, right?
Yeah.
So could you tell us a little bit more about those 17 groups?
So among the 17 groups that we found, there was like members of the Phypia genus that we know it was isolated from freshwater systems, and it utilized as a salt carbon source dimethyl sulfone, which is an intermediary, as I said, of the DMSO oxidation, which is one of the most abundant, is very abundant in the atmosphere in the area that there's sulfur. So we found a lot of DMSO there. So we know that when it oxidizes, bacteria can utilize these compounds and then use it to grow and metabolize. So that was very interesting. And also, we found members of the Oxalobacter family that utilize also oxalic acid, which is one of the most abundant decarboxylic acid in the atmosphere, which we're seeing like a thread of organisms that can utilize things that are present in the clouds and in the atmosphere that can allow them to probably stay there. But those are questions that we need to.
So they're not just floating around; they're actually living there, and I think
There's
that's
a
what's so
suggestion.
exciting.
Yeah.
So let me add my perspective here. So we don't have evidence to suggest that they are metabolically active at these conditions, very high in the atmosphere, very low temperatures. But the other aspect here is what seems to us, the humans, extreme may not be extreme for them. For example, as you know, we store them in the lab at -80, and that's how we keep them, as stock solutions. So it's at least theoretically possible that they can be metabolically active, but we haven't really proved that. And so that's one of the next things we're going to do. And we do it a couple different ways. So we now want to go and sample, again, with aeroplane in situ, but have more online pipeline that can tell us if they are metabolically active. And the other way, another research we are doing is, together with our collaborators at Georgia Tech, we have a nice atmospheric chamber facility. So we have basically a chamber that can simulate the conditions in the clouds. And we want to put our bacteria, a few that we isolate from these samples, and see how they behave. So I think, as you guys said, and I think I say too, is this opens up a lot of very exciting lines of research, and we hope to continue, and maybe in a couple of years have a more complete understanding of are they contributing to clouds, how much.
I can also imagine that clouds might have microenvironments. In other words, in microbial ecology, the world is not a petri dish. And even on a petri dish, the edges are a little bit different than the middle. But I can imagine when you see these immense cloud formations, and you even see movies of how they form, that there are probably little pockets that have their own little microenvironment. And it may be that there are places where the bacteria can actually grow for a burst of time and then go quiescent. And then, when more nutrients get washed in, kind of another burst. So I suspect that we'll be learning a lot of new ways of survival and persistence that maybe your cloud chambers can show us.
Yeah, hopefully.
So one of the things that you mentioned in terms of metabolism that's very interesting, maybe not everyone in our audience realizes, is the importance of sulfur compounds produced by marine organisms and their predicted role in cloud formation and rain. And in contrast to what's happening really high in terms of ice formation, could you give us a little perspective on that?
In the case of the sulfur area, we're not like the expert part in the atmospheric part, but in the high altitudes, what we're seeing is that it's not only sulfur that we find in the very high altitude, but more like dust and things like that that are less likely to form cloud droplets, but that form the ice crystals. So at high altitude, what we see is these ice crystals forming that are formed by dust, pollen, bacterial cells, and things like that. But at low altitude, we saw more like these sulfur compounds that are more likely to form cloud droplets and CCN. So in the case of, I think bacterial cells will be influencing more the mixed phase clouds in the part that there's like more ice forming clouds more than the CCN or the cloud condensation nuclei or water droplets that are forming at lower altitudes.
So let me see if I,
Yeah.
let me repeat this, and
Yeah.
you tell me if I've got it right.
Yeah.
At the high altitudes, it really is the ice nucleation that plays such a key
key
role
role,
here,
yes.
because you need a critical size at that temperature and pressure
Yes.
to actually be involved. And at the lower elevations, in fact, the microbes in the ocean that produce a lot of
The DMS.
DMS that makes it into the lower level of clouds can stimulate rainfall.
Yes.
And so you really have something very different going on at the two levels, but both profoundly influenced by microbes.
Yes.
Right?
I think it's a little stretch to say profoundly
Oh.
impacted yet. But we think they play a significant role, and I think that's the challenge, to figure out how significant, get some quantitative data about that. So I'm a little skeptical to not say yet profound impact.
Yeah.
Absolutely
Yeah.
right. I use profound in the sense that it's profoundly
Yeah.
surprising to me that this would have been true.
Yeah.
But again, I think this is probably a better question for our collaborators, who are the atmospheric chemists, what is going on in terms of the inorganic aspect, and we are looking more on the biological. But the other thing I know from them is the clouds in the upper troposphere, they are cirrus clouds, as they call them, and they are more important for the climate change, because I think they reflect more of the UV radiation, and so I think it's the unknown part on the climate modeling and how the atmosphere works. So that's why we are focusing also on them, because it's a little more unknown what's happening up there, and they are more important for the climate.
This just reminds me, too, of how microbiology is really poised at interdisciplinary facets in research. I think Jeff Miller's president's remarks mentioned that yesterday, and I think that as we train scientists, we need to think about more opportunities. And I think Natasha's very lucky to be in a group where you're naturally collaborating, and that the institution where you are is known for those kind of collaborations. And I think that's something that we need to keep in mind when we train students, and grant makers need to keep in mind when they fund people, is to really try to enhance those interactions. Because I do think you're right. We have a lot to learn, but I think we can only learn if everybody contributes from their own specialty. I think it's a very exciting time for microbiology in general, and this is one of the examples that reminds us how exciting it is.
I fully agree, 100%. I mean, for example, in Natasha's case, now she studies the microbes in the clouds, right? But she needs to know bioinformatics too, because the data, the genomic data and the metagenomes we are working now, they need bioinformatics skills to analyze and see who is there, what kind of genes they have, which genes are rich in the organisms in the atmosphere compared to the soil or the ocean, so we can figure out what are the adaptations, how they resist the UV or the little water around. So, if you think about you studying the microbes up in the atmosphere and you still need informatics, so I think that's a great example where, we need to train the students to be more, trained in different aspects, not only the traditional classical microbiology, which is very important, but yeah.
I think there's another aspect to this, and that is when you think of the cost involved in these flights and the analysis of data involved, the more scientific information from all aspects that we can acquire, the more it makes it a great investment for our government and the taxpayers. And so this is really a nice example of that. I think that there's one other thing that comes back and ties into this interdisciplinarity. Microbiology has always been a very interdisciplinary field. But as you said, metagenomics has just opened the doors for us to be able to ask questions that we couldn't ask very easily before. And that's why this metagenomics and bioinformatics, coupled with analysis of so many different environments, has just opened our eyes to the fact that microbes drive everything on an earth.
I think also, even though we're studying the present day, what we learn may help us to reflect back on days of the early Earth. And also, if we're doing interplanetary exploration, many of the planets in our solar system and beyond are quite vaporous. And the idea of studying life in the vapor phase is not anything that most of us in our regular laboratories do. So you may also have an impact on astrobiology and the kinds of investigations that take place for us to look for. You know, what we look for when we look for signs of life elsewhere? First, we look for water, any form of water. And so this form of water is a new medium for us, and I think it's very exciting, the light you may shed on how bugs grow there.
I think another important aspect of this work is how microbes transfer across continents. Because the thing is, if a microbe is found itself at 8 to 10 kilometers, the winds are so strong there that it can go around Earth in a couple weeks. That's why when you fly, you have this jet stream that is, 50 miles or 60 miles per hour. So they can travel around very quickly. So it's interesting to see which microbes make it up there. And that's another line that we pursue. It's because not all of them make it there. And we have some hypotheses why we think some make it and some don't make it. And we're going to test it with our chamber, etc. But I think it's quite exciting line of research to see which ones make it up there, what cell properties do that, and then how this correlates with how they transmit. And it's interesting, like in one of our samples, or two, from the hurricanes, we found fecal bacteria at these altitudes, but not in the others, not in the background or before the hurricanes, which suggests that maybe even pathogens could move this way. And so I think that's another important aspect, not just the connection with the clouds, but also for biogeography. And so again, I think we go into the interdisciplinary type of research.
So that's an area that has been really studied nicely with respect to fungi and transmission of fungi around the world associated with the upper atmosphere. But when you're getting as high as you are, you're not seeing as many fungi as bacteria. Is that correct?
Yes. Yeah, we saw like on an order of magnitude less fungi than bacteria in the upper troposphere.
An order of magnitude less is still a substantial number?
Yeah, it's still
Right.
a lot, but not as much as bacteria, as you can see in the lower troposphere of fungi.
Right. No, it's an interesting question because you might expect that if, let's say, for example, a virus. You haven't looked at viruses yet,
No.
although presumably you could pull some of them out of your metagenomic samples.
Yeah.
It would be interesting to see. The predictions you'd make about virus resistance to UV and resistance to a lot of these environmental conditions, you might think that they would be the first things to be transmitted widely around the world. But we don't see that as much based upon the epidemiology of transmission of viral diseases.
I don't know, we haven't looked at viruses yet, but what we can say with confidence, I think the cell size plays a role. Like, for example, we don't see as much fungi you see typically in lower altitudes at the high altitudes, and we think it's because of their size being significantly bigger. So Natasha, when she did her work, look at the microscope, under the microscope, some of the samples, it seems like all the cells are between, I would say, a couple hundred nanometers and 1 micrometer. And usually fungi are larger than 3 micrometers in size. So I think the size plays a role because the higher the size, probably the easier are for them with gravity to sediment. And so I think that plays a role here too. And maybe viruses are maybe too small, I don't know. But we haven't looked at viruses yet.
You know, another interesting thing is to think about metabolism and what you said earlier about how you assay whether they're really viable and metabolically active in those areas. That's the one disadvantage we have with metagenomics is we're looking at sequences instead of activity. So if you were going to choose using an assay in the airplane to try to detect metabolic activity, what would you do? What would your assay be?
Okay, we are I would say we are in the initial stages of trying to design that. But we were thinking maybe to target some of the key molecules, like for example ATP. So basically to see under atmospheric conditions, either in our chamber or if we can do it online, real time in the aircraft, if we can see ATP production, ATP signal and things like that. So that's something we are thinking as a first kind of idea. Because ATP is a characteristic molecule of life. We don't see it in inorganic particles. And so if we see some change in ATP content, that is indicative that something is going on.
It'd be interesting to measure the redox conditions in the cloud. That might also be an interesting symptom. And then you might know what kinds of reactions might be possible too.
Yes, that's very difficult to do in situ, but maybe with a chamber and our collaborators, maybe we will be able to manipulate these things, because the chamber can do that. They can change the content of oxygen, concentration of oxygen. Let's see.
It's exciting.
But the exciting thing here is that you're thinking about assays that don't exist.
Right.
You're thinking about looking at things that we didn't know about before. And so I think that for all the students in the audience, I think that this is really exciting to think about the future and what you can do, what you can discover. Right? So it's very exciting. Now, Natasha, you're a graduate student right now. Where are you in your career?
I'm hopefully close to graduation at one point. Yeah.
And then what are you planning on doing?
I planning to keep working in some of these atmospheric microbiology interactions as a postdoc, where I found one.
So you've been a pioneer so far. You're going to keep that up, right?
Yeah. Hopefully I will planning to become part of the academia at one point, to keep doing some interdisciplinary research, and I think it's so interesting and so important to make new discoveries in science.
Well, this has been really exciting, and I think that it ties into discoveries in places you would never have imagined before, and also ties into the fact that microbes are everywhere. There's some kind of water, and there are so many things you can imagine to do in the future. So it's an exciting time to be a microbiologist. Thank you very much for joining us today.
Thank you for having
We
us.
want to thank.
Thank you for having us.
Yeah.
Thank you. We want to thank the ASM Live audience, both those of you who are here in person and all of you who are online. Thank you very much. Please keep joining in to hear more stories from the ASM Talk Show.
Extending the legacy of a classic reference, EcoSal Plus joins the ASM family of periodicals this June. This growing collection of continually published reviews reveals our ever-expanding understanding of the biology of E. coli, Salmonella, and the other Enterobacteriaceae. From cell architecture and growth to pathogenesis of infections to life in communities, EcoSal Plus is the single source of knowledge about these organisms and their use as model microbes. A distinguished editorial board, led by Editor-in-Chief James Caper, ensures that content serves the needs of the research community. Free trials available for institutions. Take a look at ecosalplus.asmblog.org.